Hi, everybody. Welcome back to the next episode of You Don't Have to Struggle. I'm Dr. Lee Tannenbaum.
And I'm Lisa Tannenbaum. This is the beginning of our third season, and we have upgraded some of our technology. And we're now coming to you from different remote locations. It does enable us to have a very exciting guest today. Dr. Tannenbaum, you want to introduce our guest?
Yes, today Dr. Arman Shekari has graciously agreed to be our guest. Dr. Shekari is a PhD in neuroscience and a lecturer at Trent University and McMaster University in Ontario. Dr. Shekari is actually a really smart person who knows a lot about neurochemistry. Lisa and I understand how to treat patients with addiction, and then we hit up against a wall of hard science and we pretend to know what goes on beyond that wall. But Dr. Shekari actually knows what's going on the other side of the wall. As Lisa noted, we're all from remote locations today. I'm at our studios in our secret location here at the Buttercup Studios. Lisa apparently is in some cave somewhere where she doesn't have good lighting. And Dr. Shekari is in Toronto. We have international guests, which is also a first for us. So welcome to the show, Dr. Shekari. Thank you so much for being with us.
Lee and Lisa, thank you so much for having me. You set the bar pretty high for me. I'll try to disappoint your guests.
That's probably pretty hard to do, so I think you're going to be fine. So the first thing we asked Dr. Shekari about was the GLP-1 agonist that Lisa and I have spoken about several times. These are the medications that many people are familiar with because they're being used very aggressively right now for weight loss, but there's also been some crossover where they are being used for addictive disorders. Dr. Shekari, tell us what you know.
Sure. These are, if you kind of got to be under a rock if you don't know what these are. Ozempic and the like. They're GLP-1 agonists, like you said, Lee. It's interesting because addiction and a lot of the pathways in the brain that are involved in addiction, they evolved on a background of just food. That's kind of where it all comes from. Back in the day, the really only kind of big dopamine rush you got was when you consumed high calorie food. That is the evolutionary background with which the circuitry in our brains that kind of modulate reward, that's where they come from. So the idea that a drug that is involved in, satiety and suppressing your appetite, that it can be used for other types of addiction, when you kind of look at the evolutionary background of what's going on in the brain, it's not super surprising that it can have other more broad effects in that way.
One of the things that I'm sure you remember and are aware of is this talk when GLP-1s first started getting big. And for anybody who missed it, we're talking about the GLP-1s are Ozempic, Wegovy, Zepbound, these new weight loss injections. When they started getting big, the big topic of conversation was that people were having their food noise disappear for the first time that they could remember. To Dr. Tannenbaum and I watching it, we know exactly what that is. They're using the term food noise, but it's cravings. We might as well call it drug noise when it's used in a drug situation. So that was the first sort of clue that these systems were closely enough interrelated that the suppression would overlap.
Yeah, for sure. That's an interesting point you bring up because these drugs, mechanistically, and also you talk to people anecdotally, they suppress the cravings, but importantly, they don't suppress the liking. So they don't cause anhedonia, which is really important for kind of quality of life. It'd be really bad if sure, you're not eating anything and you're losing weight, but you don't really derive joy from the things that you do eat because these drugs don't, eliminate your appetite. You're still eating some things and you'd like to enjoy those things. But thankfully, because of the way these drugs work, they don't really mess with the liking, which is really kind of separate from the wanting or the craving or just the kind of drive to get these things. That's where they kind of target, which makes these drugs really popular and also much more tolerable with respect to how they deal with your quality of life.
Do we know what receptors they're binding in the brain? Are they binding receptors?
They are. These drugs, I know they've kind of been like a big splash in the last, 3-4 years, but we've known about the GLP pathway for decades. And GLP-1s, there was research in the early 2000s by the company, I think, that's now Novo Nordisk. So what they do, there's kind of like the boring stuff, and then there's the interesting stuff that has to do with addiction. The boring stuff is how they're also a diabetes drug. So what they do is they go and they bind to these receptors. When you eat something, your intestines release these things. They're called incretin hormones. One of them is called GLP-1, or glucagon-like peptide 1. The other one is a really long name. It's GIP. I think it's glucose-dependent insulinotropic polypeptide. And tirzepatide actually is an agonist for both. That's like the new drug from Eli Lilly that's like really good at weight loss. But a lot of them just do the one, which is GLP-1. When you're full and your intestines release this, what this does is it goes into your pancreas where it binds to receptors on the cells in the pancreas to release insulin so that, you can intake the glucose into your cells, and that's been known for a long time. It's pretty straightforward. It's very good at that. It's very good for people who have type 2 diabetes. What it also does is it goes up into your brain and does a few things. Some things related to satiety, so it goes up into your hindbrain. So that's the back part, the most primitive part of the brain, and it hits a few areas. One of them is called the area postrema, or AP. This area has a bunch of neurons in it that are involved in both satiety and also aversion, like nausea, and they kind of overlap. That's why in some people, especially in the beginning, these drugs can cause some off effects or side effects like nausea, in addition to satiety, which is a little uncomfortable for people. But that area in the back of the brain is actually pretty leaky with respect to the blood-brain barrier, and that's on purpose, so that these metabolic peptides can get access to the brain to let you notice you're full, stop eating. And that's the basic, almost like reflexic part of these. But they also go into the midbrain, and it's here where magic happens. They hit the VTA, or ventral tegmental area, and the nucleus
We're very
accumbens.
familiar with the VTA.
We love the VTA.
There you go. And so do a lot of drugs. Right? That's the classic dopaminergic access that is involved in regular cravings, but then drugs kind of hijack this, take it up to 11. They do bind to receptors here too. And again, this is on purpose. This is the kind of signal that your brain gives to like, hey, you've eaten something. This is rewarding. Okay, good. But now you should chill out and relax, and you shouldn't have cravings for at least a little while, because you're full, you've eaten. Back in the day, again, remember evolutionarily, getting a high calorie meal was a really risky thing to do. You'd have to hunt an animal. You'd have to be very motivated to do this. And then once you do it, it's probably in your best interest to go chill out. I mean, when you eat a big meal, what's the first thing you want to do is you want to go take a nap, right? Just sleep and not do anything.
This is the rest and digest of the parasympathetic nervous system. There's a whole skeleton structure through all the bodily systems that are focused around this idea of first you expend a lot of energy, and then you stop your energy expenditure and you focus on absorbing what you got.
That's right. A lot of that is physiological to the parasympathetic nervous system, but a lot of that also, because we're humans and we're smart and we have big brains, it's got to be kind of psychological, or I don't know, neurological, if you'd say. And it does this, these GLP-1s activate receptors in the VTA in a type of neuron called a GABAergic interneuron. What a GABA interneuron is these are neurons that kind of turn things off. So they turn off the dopaminergic neurons in the VTA, so they reduce the amount of dopamine going to the nucleus accumbens. One of the ways they kind of tamp down that dopamine. But another really interesting thing they do is they act on neurons that are coming from your prefrontal cortex. That's the opposite of the hindbrain. That's the most advanced part of your brain. From back to front, you go from least complicated to most complicated. These neurons go in to the nucleus accumbens, and they are involved in things like impulse control and behavioral control, things like that. And GLP-1s also act on these receptors to increase the amount of glutamate, which is a neurotransmitter. You bring those up, and that increases the connection between your prefrontal cortex and your nucleus accumbens. And this helps you modulate your behavior. And in addiction, this connection between these two parts of the brain are severed. So there's not a lot of talk between the prefrontal cortex and the nucleus accumbens. So you act on impulse. And the cool thing about these GLP-1s is they rescue, or they can reconnect those two parts of the brain through things we'll talk about later with epigenetics. But these drugs are designed, and this might be a good segue for you, Lisa, to talk about how we went from mechanisms to these long-lasting drugs. They're designed in a certain way so they kind of stay in your body for a really long time. So I believe Ozempic is like 85% similar to like your natural GLP-1, but there are a few key amino acid changes there in the structure which help your body not break it down. So it stays in your body for much longer than your standard GLP-1, so it can have a long-lasting effect. A lot of these drugs are injectables. You don't want to be injecting every 4 hours. The bigger the interval, the better in some cases, as long as you can tolerate it. But in a nutshell, yeah, that's how these things work.
So I'm going to override you, Lisa, because I have more interesting questions to ask.
You don't have a question that's as interesting as my fun fact.
I'm sure I do. Go ahead. Get your fun fact in.
Really quickly, my fun fact.
I'm curious to know too.
Is, and you can correct me, Arman, if you've heard something different from what I have read, but in doing a little research into the history, as you said, we've had information about these peptides for decades. And we had, I want to say, exenatide was the first one, or one of the first ones that was starting to be given to patients. But as you said, it was way too fast acting. Your body's producing these things endogenously. When we give them to you externally, it's gone. It's not useful. It doesn't stick around. And the big change, the big breakthrough came from getting the venom of a Gila monster, which is a lizard that can go for weeks and months without eating. And I might have been a scientist in Toronto, if I'm not mistaken, or in Ontario, who spoke to a zoo in Arizona in the US and had them send a Gila monster that they could study. And it was by copying some of the structures of the Gila monster venom that they were able to prolong the life of these peptides to make them useful.
Okay, you're
Sounds
right.
familiar.
That was an interesting fact.
That's a very fun fact. All right, now you go ahead with your questions.
Tying all the mechanisms of the brain together like you just did, and Lisa and I often talk about the ventral tegmental area as being the root of cravings and the lack of ability of the prefrontal cortex to affect the processes in the ventral tegmental area. Two questions. One, would you consider obesity and overeating primarily a brain disease, which would be primarily an addictive disorder, as opposed to a metabolic disease, as it's always been previously considered? And two, do you think that the mechanism of the GLP-1s would ultimately at some point maybe provide us with the ideal anti-addiction drug if we can have an effect across these various areas of the brain and how they communicate with each other?
Those are two really good questions. I'll start with the first one. Unfortunately, the answer is kind of boring. I'm not going to pick a side. It's both, because your brain and your body, they talk to each other. And when it comes to obesity, the crosstalk is actually really sinister. I know this because I've looked at the mechanism. I also have a personal connection to this because when I was in graduate school, did not take care of myself. I didn't exercise. I ate really poorly. And I was 50 pounds heavier than I am today. And I remember when I was like starting to lose weight and realize, what I was doing to myself, how strong the cravings were for me. And it's because when you lose weight, when like fat exits your body, fun fact, a lot of the fat that you lose, you actually end up breathing it out. That's the kind of final form it takes when it exits your body. But when you're losing fat, the actual cells, the adipose tissue in your body that stores that fat, those stick around. They shrink, which is why you shrink. But the actual cell itself hangs out. And that's a cell, it's a part of your body. It's connected to the vasculature. And when it's really small, it's not happy. Remember, the entire circuitry of addiction is all, it's evolved around food. So when you're hungry, your body is not happy. And there are many, 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 many redundant pathways that you have both in your brain and in your body that really want you to never be hungry and always want you to have like as much excess energy in your body as possible. So when they shrink, your body kind of goes in the crisis mode and it just starts dumping these hormones into your bloodstream that increase cravings like leptin and the such. And it just really has a hold on you. And when you start losing weight, that's why a lot of these diets are they call yo-yo diets, up and down. You lose the weight, your body gets upset at you and you bounce right back. And it's because these cells are still around and they're talking to your body and they're also talking to your brain to say, go and eat. And fortunately, they do end up dying eventually and kind of acquiescing and going away. But it takes like 10 years for them to go away. So think about somebody, yes, you can lose 20, 30 pounds in a matter of months, but it takes a decade plus in some people, it depends on your genetics, to really get rid of these cells so they stop talking to your brain. I still have cravings for a ton of garbage. I have a sweet tooth. I think it's genetic. But I remember how powerful they were and they last for years. So if you want to lose weight, you need an intervention. For me, I always say it has to be sustainable because you got to be able to do it for years. Once you lose the weight, you're not done. You have to get rid of that adipose tissue, for good. And that takes a really, really long time. But drugs like these GLP-1s, they really help because they go and skip all this stuff and they go right to the VTA and they just go, shh, calm down. Don't worry about it. Suppress that dopamine. And they help you kind of get over the hump. And if you can be on them for years, that can help the metabolics do their thing. And then you can kind of get over the hump and live that sustainable lifestyle. But if you go down that path where, your metabolism is just kind of out of whack, you can't dig out of the hole. So these drugs really help you dig out of the hole, but it's up to you to then be sustainable after the fact. So it's a brain-body connection. So I think it's both. Your second question about these drugs, eventually getting to the point where we can have a perfect addiction drug. Yes and no. We're seeing this now actually with the clinical trials that are out for these GLP-1s. We know they're good for food addiction. That's easy. They're really good for alcohol use disorder. They're excellent for that. You know that. Nicotine, And cocaine, actually, you'd think they're really good for that. Actually, we're seeing moderate effectiveness. These are very early too. These are preclinical. We think that, there's more going on there. But what they're not good for, and I'm sure, Lee, you'll appreciate this, is opioids. They show mixed effectiveness for opioids because it's a totally different pathway. Those same neurons that the GLP-1s act on in the VTA, those neurons also have mu-opioid receptors, which are trying to turn them on. And then you have Ozempic trying to turn it off. So they're fighting with each other. So there's that overlap, and all the analgesic effects of the opiates, that relief that you get when you take these drugs, that's different parts in the midbrain and also a lot in your spinal cord. GLP-1s aren't doing anything over there. So they're not touching those. You still have those pathways happening. So because there's not a lot of overlap, and the ones that are kind of dual, these drugs have different mechanisms of action. Is there a universal thing? Maybe, but the GLP-1s kind of aren't there yet. And when they do work, they help with the craving side of things. But the actual blunting of the effects of the drug, like they do with food, and it seems like with alcohol and maybe a little bit with nicotine, not all drugs work that way. So it's kind of a mixed bag.
I absolutely love everything that you said. I think that's really fascinating with the adipose cells. As you said, and as we've said before, the mechanism of opioid drugs is a different pathway than these appetites that we're talking about with the GLP-1s. The natural history of this disease course that you're describing sounds to me so similar, where you build up a tolerance over time, you're consuming opioids, your usage is probably gradually increasing the way that people's junk food eating tends to increase. And even when you stop, these cells that have been building up due to this exposure over time are still there. They're still talking to your body. They don't go away. It takes years or decades for them to ever subside if they do. So this is an issue we run into with opioid treatment all the time, where people say, all right, I've been sober 2 weeks. I got through the worst withdrawal. Now I'm done. Woohoo, I'm cured. And we're like, no, you're not done. Your oversaturated opioid cells are still there. They're still talking to your brain. They're not going to go away anytime soon. This is just the beginning. And it's really interesting to hear that repeated.
Yeah, it's really fascinating and fantastic to hear the scientific background of stuff that Lisa and I kind of know. But like I said before, we hit this black box, and then we just kind of say, well, that's just the way it is. So it's really great to hear what's going on inside. Another thing that I always talk about, and I have a hard time convincing people that I work with about the long-lasting nature of addictive disorders. And when I really have to give them a lot of reasons why addictive disorders are so chronic, ultimately the last point I get to is I say, well, there's an effect on epigenetic enzymes. And that usually shuts everybody up because nobody knows what they are. And they certainly don't think they know more about it than I do. And they're not ready to argue with me about it. But that's really all I know, is that there's epigenetic enzymes which affect the way DNA is expressed, or the way proteins that are made from DNA are expressed. So even though an addictive disorder can't change your genetic information, it can affect the epigenetic enzymes, which can affect the way your genetic information is expressed. And in opioid addiction, this primarily presents itself because there's a change in the opioid receptors. And you probably know a lot more about that than I do, so take it away.
Epigenetics, it just means above genetics. Epi just means up. Huge area of really important research. It's probably the big reason why the Human Genome Project did not cure all known disease like we thought it would, because there's so many things going on. Yes, the genes that you have are important, but I honestly, as a molecular biologist, I'd argue the expression of the genes, I think, is more important than the genes you have in most cases, in a vast majority, because you think about like humans and our next common ancestor, we're 99.7% genetically identical. So between human to human, I mean, it's like 99.99999. You're right there. Yes, there are some genes that have like pretty big impacts on things, sure, but the expression of these genes by far and away is really, really important. And that's all epigenetics. And really, it's once you get into the weeds, it is really, really complicated. But for the listeners, really, it doesn't have to be. All it is you have your DNA, which is a giant string of nucleotides, and you have all these proteins in you, machinery that has to get access to these strings of letters, to these big, bulky things. And your DNA is this big, tied up, wound up mess, which is on purpose. It's beautiful, but it's really dense in there. What you got to do is you got to get that machinery to access certain genes at certain times in a certain way to make the right amount of protein in the right amount for the right amount of time. So it's a really complicated dance. And what your body does is very basically makes the access of these genes either easier or harder depending on the environmental circumstance. So it can do this by adding things like methyl groups or methylation, which is just this kind of bulky chemical thing that makes it harder, usually, for certain things called transcription factors, which are the machinery that kind of comes in and makes the RNA, which becomes the protein, makes it harder for them to kind of get to where they need to go. Another big one, they're called HATs or HDACs. These are histone acetylases and deacetylases. Acetylation is something that, another kind of chemical thing, but it's the opposite of methylation usually. When you add it, the genes around the DNA structure relax. They open up and it makes it more accessible for these things to access them. And what we see with addiction is certain genes actually end up being overexpressed. So there's too much acetylation happening at certain sites. So these things are aberrantly expressed. One that I know about is a brain-derived neurotrophic factor. This is a big plasticity marker for your brain. The more of this you have, usually it's good. It makes you smarter. It increases the amount of connections your brain can make. But in the context of addiction in the VTA, when you boost your BDNF too much because there's too much expression happening, that's not good because you physically rewire your VTA in a way that makes it hypersensitive. And that's permanent because BDNF creates lasting structural changes in your brain that stay with you. And you're doing this in a drug-dependent context. So even if, like Lisa said, you get past that initial withdrawal, which is very bad physiologically, those networks are with you for basically forever. And then any kind of cue or a relapse or anything immediately activates them. And you're right back where you started, where someone who's doesn't have an addicted brain, let's say, they don't have those networks to start. It's much easier to just fall back down the rabbit hole because some of these genes that are overexpressed physically rewire your brain to make it much easier to fall into the kind of grips of addiction.
So would it be a useful analogy to say that these plasticity markers are like being able to access the wiring in your car, where in some circumstances it's really helpful if you need to, you can jump your battery or you can fix an issue with your car, but also it makes it way easier for someone to steal your car?
That's exactly right. And you bring up a really good point in that all these things that I'm talking about, they're not by accident. These are built-in things that your brain and body have. They serve a purpose. Another example I'll give you is this thing called delta phos-B. It's a transcription factor, but it's really weird because most of these transcription factors, they decay or go away in a matter of, 30 minutes to an hour. You don't want them around for too long, kind of like with the GLP-1s we're talking about. This one lasts for months and months because it's important in chronic stress. When you're under chronic stress, delta phos-B accumulates in a nucleus accumbens, and it's there on purpose because it's to make you more resilient. When animals are under chronic stress, like social defeat, I think social defeat's the big one that's been studied over and over again. And the more delta phos-B they have, the more resilient they are. The more likely they are to bounce back and try again versus kind of give up and withdraw and become depressed. Unfortunately, when you take drugs, that delta phos-B pathway, it's just increased so much from baseline. It's like the ultimate chronic stressor that now, instead of strengthening, what it does is it weakens the negative affect pathways. So you're blunted from negative emotions so you can keep going. But now what it does with drugs, it's so much higher, it goes all the way around and it strengthens the positive pathways in the nucleus accumbens. So now you're hypersensitive to positive stuff, which makes you much more susceptible to falling back into addiction. And it's a double-edged sword. People with higher delta phos-B are much less likely to suffer from things like major depressive disorder, but they are much more likely to fall into the grips of addiction. These are the go-getters. So they want to go get, whatever it is, whether it's success or whether it's illicit substances. So it's a double-edged sword, and a lot of it is just epigenetics.
And that's really fascinating because we've talked about this before. So many of our patients that we see clinically have been misdiagnosed with things like depressive disorder, when in actuality, what they're experiencing is this rapidly cycling addictive trend.
It's also interesting because we've talked about how a genetic advantage of being able to process a high-calorie diet was a genetic advantage to get us to this point in our evolution. But in America, where we have so much food available so easily, it's become a disadvantage. And we see the same thing with a lot of our patients, that we don't have the type of chronic stress that we were evolutionarily built to withstand, like hunger and cold and the animals chasing us down. But we have we have this artificial chronic stress that we've created with the news media and our phones and the emails and the constant communication and the feeling like I have the stress of what's happening in Ukraine, like it's happening next door to me. So we have this never-ending, ongoing chronic stress, and certainly that sounds like that would affect our behaviors. But the question I wanted to ask you was, are any of these mechanisms that you know of currently the target of anybody trying to pharmacologically affect them?
So I just want to point out, this is not supported by trials at all, but this is something, Dr. Tannenbaum, that you and I talk about anecdotally all the time. We observe anecdotally that our patients who are under overwhelming chronic stress, both emotionally and neurologically all the time, are very, very, very resilient to the things that would make you or I absolutely just pack it up and call it a day. They weather problems in their life that would be, the only thing that we talked about for years. So it's really interesting.
They function very well. They don't function well in a functioning world, but when the world becomes very dysfunctional, they actually function pretty well.
They do great.
They're way better than I would function in a dysfunctional world. So yeah, that's really fascinating.
Delta phosphate. That's probably a big thing that's happening there. I mean, that honestly, like when you try to understand the underpinnings, like, hey, what's happening in the brain and the body with people who are suffering from drug addiction? It makes them human beings. Now, if you don't understand, you see this, you can't understand this behavior. I would never do that. I'd never do this. I can't even believe they're doing this, that, and the other. No, it's a disease, and your brain, your physical, structure in your body is different. It's different as we're talking about epigenetics, in a pretty lasting way that's hard to reverse. So humanizes it a bit, and you have sympathy, and that's really what you need in this space, because usually these people end up in prison, and it's terrible. They need help. They need systemic, long-lasting help, because what's happening in their brains is systemic and long-lasting. So understanding what's going on and doing what we're doing, talking about it, trying to educate everyday people, it's really important to help reduce that stigma, because really you see what they're doing is adaptive for their situation. Their brains are just responding to what's in front of them. Your brain is really, really good at that. Every single human being's brain is. Now, to your question, Lee, are there people who are trying to target this for some kind of therapeutic potential? Yes, it's very early. There are things called these HDAC inhibitors, so histone deacetylase inhibitors. So these are things that will help get rid of those acetylation groups to kind of tighten that DNA back up so it's not being expressed too much. That's tough to do. It's very messy because your entire body is regulated by epigenetics, and that's too broad of a brush you're painting with there. But I know there is a lab, I believe her name is Elizabeth Heller. What she's looking at is trying to use targeted genetic tools like CRISPR-Cas9, where you can actually take these designer things where you can go in and target a specific gene, go in and actually target not only specific genes, but specific genes in specific areas of your body. So the nucleus accumbens, for example, you can target BDNF only in the nucleus accumbens and get its expression to go down. You don't want BDNF to go down in the rest of your brain because it's a highly, highly neuroprotective thing. It's really highly associated with intelligence. So you don't want to bring it down everywhere. But if you can tone it down a little bit in that specific area of the brain, that's great. This is still in the lab, still in Petri dishes and stuff. So it's a long ways away, but we're starting to look at that like, hey, we know what the targets are. We need really fine control because we're in the brain. Like I told you, with Ozempic in the hindbrain, the same neurons in the back of the brain involved in satiety, they make you nauseous and want to throw up. So you're really careful with how you work with things in there. We're working on that selectivity, but it's going to take a really long time.
Not the place for a shotgun approach.
No.
We're big sci-fi fans, and I'm just picturing the dystopian future in 100 years where you pull up an app on your iPad and you can micro tweak the settings of acetylation in your own brain in different areas. That's going to be a really interesting world.
Let me move on to the last topic of the day. I know you had brought up that you had done a lot of work with glial cells, and glial cells make up the sheath of tissue that surrounds our neurons, which we always talk about neurons. That's the workhorse of the brain. And I have always just known that when neurons become coated with glial cells, they become faster, and therefore they become more efficient neurons. But apparently there's other mechanisms of how glial cells can play a role in addiction, which I'd love to learn about.
What you're thinking about are the oligodendrocytes. These are the cells that form the myelin sheath. around the axon. And there's two different other types of main glial cells. You have the astrocytes, which are kind of like the helpers that kind of do all the stuff the neurons aren't great at. And then you have the microglia. Microglia are the immune cells of the brain. Your brain, everyone's brain, is basically cut off from the rest of the immune system. It's got its own immune system, and it's the microglia mainly that kind of drive that. When you take high doses of drugs, that your brain sees that as a big chronic stress or a problem, and what it does is it upregulates an inflammatory response. That's not good if you do that long term, because what these microglia can do is they can actually go in, much like with BDNF that we talked about, and they can change the structure of the circuitry in your brain. They can. It's not just neurons. Neurons are cool. I mean, I've worked with them for many, many years. Glia, like you said, people just think of them as their support. No, they're really, really, really important. Like, if there's no glia, there's no brain. So now we're just kind of starting to appreciate the role they play. A lot of these therapies that only target neurons, sometimes you take them to clinical trial, they kind of disappoint. Well, you've ignored the major other cell type in the brain. You haven't even paid it any mind. But in the case of these microglia, they break these connections in some areas of the brain. A big one is the striatum, which I'm sure you know about. It's involved in habit formation, goals, reasoning, decision-making, all this stuff. And when you get rid of these connections, what you're doing is you're creating basically inflexible thinking. So you're not having somebody be able to be flexible with the way that they think about their goals and behaviors. And this is because their brain is chronically inflamed and it's pruning away all these connections in areas like the striatum. And that's all glial cell derived. Neurons are just kind of sitting there and taking it. That's one pathway I know about that we can discuss.
When you say that the brain reacts to heavy drug use with an inflammatory response, does that have to do with the fact that it's literally an exogenous substance coming from the outside, or does it have to do more with the amount and the constant stimulation? Do you know what causes that
more
inflammation?
The second one.
It's more
the second one, yeah. Because the reason these drugs work is because they are mimicking endogenous substances. So if they didn't work and there was just this foreign thing, your body would just attack it. It wouldn't even make it to the brain. Not to say that doesn't have some kind of impact. I'm actually not an expert on that, but I do know because of the massive, very abnormal impact that these drugs have on your brain, they cause a lot of things. Excitotoxicity is a big one where your brain gets too excited and it starts kind of like dying, which is not good. This creates inflammation. And unfortunately, when this happens, it does things in your brain where it's opposite of what should happen. So when you take a drug, you massively increase the amount of glutamate in your brain. There's this thing that's on your glia, on your astrocytes called GLT1, glutamate transporter 1, a vacuum for glutamate. When there's a lot of it, it kind of sucks it up, cleans things up. If there's a ton of glutamate happening in your brain, you'd think you want more of that thing. But when your brain is inflamed, it actually downregulates the amount of GLT1. So you're losing those vacuums. So it never gets sucked back in. It's a paradoxical thing. So the inflammatory response that you see in the brain, which is largely driven by glia, has these negative impacts on your brain by working on other glia, like the astrocytes expressed in GLT1. You don't want to reduce that. You want it to go up. And that messes with the glutamate homeostasis in your brain, which is called a glutamate homeostasis hypothesis, a big hypothesis that we have about what happens in the addicted brain, big picture, which keeps it in this vicious cycle state where they can't break out of the patterns they have.
That's just fascinating stuff. I love this stuff. I could do this all day. Lisa and I could do this all day. But we're running out of time here on our segment. So I want to thank you once again for coming to talk with us. I hope that you'll come back again when we have more questions. And I think you've been a great source of information, definitely to Lisa and I, and I'm sure to our listeners. But I think we'll call it a session for today. And once again, thank you so much. And anything to add, Lisa or Dr. Shekari?
As Dr. Tannenbaum said, man, I'm going to go read about the glutamate homeostatic hypothesis now. I'm so excited to hear about this. If anyone out there is as excited by hearing about glutamate as we are, let us know and let us know if you have any questions. And yeah, thank you so much for coming on.
Yeah, and
Lisa,
probably the rest of you.
Me.
You don't want to be around for the rest of this discussion because this could go on till midnight because we love neurochemistry. Once again, thank you for being on our show. And to our guests, you should remember that you don't have to struggle.